Okay, so what I'll do is all the simple questions like which are direct or something like name the smell cell or give examples. I'm skipping those questions. I'm just taking up uh, the important ones since you might have done these theory based questions before. Okay, okay, let's try the fifth one. During cooking, on adding salt to the vegetables, water is released. Name the mechanism involved. Who can answer?
Is it hypertonic solution?
Okay, so that would be the property of the solution. What would be the mechanism?
Osmosis?
Yes, correct. Okay, so I think still many of you might be having confusion regarding hypertonic. Okay, regarding mechanism or when you should say solution is hypertonic, hypotonic, or the mechanism, right? So hypo basically means low. And what is this low that we are talking of is the low solute content. So basically anything which has more water or more liquid content in it, ideally we are talking only of water, so it would be water only. So which has low solutes or more water content is called hypotonic, right? And what is hyper? Hyper means high. So what is high? The high solute content, right? Okay. So basically on adding salt to the vegetables, water is released. So what happens is suppose let's say we are taking this as the example of any vegetable, let's say A, right? And once I add salt, basically the amount of uh, solute inside will increase, right? Or you could say that it would shrink. So if this was the, yeah, so it would become something like this, right? And all the water would come out like this, right? Okay, so why does this happen is because you sprinkle salt, right, on the pan. Let me just take and okay, so suppose this is your pan, right, in which your vegetables you have, you're trying to cook them or fry them, whatever, right? And then when you add salt, if I write plus salt, so basically what you're doing is you're just sprinkling salt over it, right? So salt gets accumulated or stuck on the sides of this vegetable or whatever in the medium. So it translates to something like this. There is salt all around, right? So this medium or the uh, region outside the vegetable would become hypertonic, right? Because of salt, which means it has high solute content. And that's why osmosis would occur in this direction, which is basically from here to outside, because there is less water concentration outside, right? So it's the movement of water across the concentration gradient, and hence the vegetable would lose water and it would shrink. So it would be water outside. Okay. If you want, you can draw this diagram, or you can make it in your own words or write it in your own words for now, so that whenever you encounter osmosis related questions, you don't face the confusion that what are you supposed to say. So the mechanism usually will be either diffusion or osmosis. This is the mechanism names, right? In osmosis also you have two categories. One would be endosmosis and one would be exosmosis, right? But if you write osmosis itself should be sufficient. It's fine. Okay, and if they tell you what is the nature of the solution, nature of solution, let's say, right? For this question, your answer should be: is it hypotonic? Is it hypertonic? Or is it isotonic? Right? Okay. You can make note of this also. So nature of solution would be this, and mechanism is usually one among these, whether is it diffusion or osmosis. Okay, let's go on to the next question. Okay, so we did five. Six is similar. Okay, what is the function? Nucleus. Okay. Okay, let's do the 14th one. 14th one. Yeah, 14th and 15th. We'll do both. Okay, so name two structures which are found in plant cell but not in animal cell. You can answer.
Ma'am, one is cell walls.
Okay, and chloroplast, yes, correct. Yes, Ashka. Chloroplast is correct. Okay, so two structures which are found in plant cell but not in animal cell. You can write cell wall. Okay, should I write it here? Cell wall, chloroplast, right? Okay, and what about the 15th one then? So 15th one, yes, Aryan, you're right. The 15th one is the opposite. Name a structure and the function of uh, an organelle or a part which is found only in animal cell but not in plant cell. Okay, so first you can tell me the name, then we'll go on to structure and function. Uh, no, not arms, legs. So we are talking of cell organelles or so why did I say structure is because something like cell wall. Cell wall isn't an organelle, right? For
Ma'am,
that,
lysosomes.
huh? Yes, correct. Lysosome. Aryan vacuole, no. Vacuole is present in animal cells, but it is smaller in size, right?
Okay, ma'am.
Present in both. So lysosome is the only structure which is found in animal cell but not in plant cells. If you remember, we had discussed this when we did our slides, that why do plant cells do not need lysosomes? Does anybody remember?
Ma'am, the part which the lysosomes play in the animal cell is basically taken over by the vacuoles in the plant cell. So they don't need lysosomes.
Okay, not vacuoles. Can you try again? Yes, you are right that that function of lysosome is taken over by some other part, but it is not vacuole. Vacuole is involved only in storage, right? Okay, let's do quick recap then. Okay, so I hope you remember that plant cells have uh, two structures or like two protective barriers, right? The first one is the cell wall, right? And this one would be the cell membrane, right? So what happens is cell wall, because of this double layered protection, okay, have I got the answer? Cell does the job, no. Cell doesn't do the job, the answer would be cell wall does the job. So cell wall does the
Yes,
job
mom,
of.
that was a typo. Cell wall. Sorry.
Acha, okay, okay. Yes, cell wall does the job. Correct. So what happens is because of this double layer barrier or double protection which the plant cell has, the entry of foreign particles is really very very less, right? Versus in animal cells, what happens is we only have a cell membrane, right? We have a single layered protection, so things can pass in and out easily. But in this case, the cell wall is quite rigid, and only a specific particles are allowed to pass to and fro, right? Okay, so before we come back to what cell wall exactly does, let's go back to the question. And who can tell me what is the function of lysosome? Nobody, you forgot lysosome? Acha, okay. If you don't remember the function, do you remember the other name for lysosome? Okay, yes, Aryan gave me an answer, correct. Anybody else? Lysosomes are called as what? Like every organelle has been given a name, right? Like nucleus is the controller. Yes, Aryan. Yes, Sujal. Correct. Right? So basically, lysosomes are called the. Yes, yes, Ashka. Suicide bags of the cell, right? Or they are also called the waste disposal crew, something like that, right? Waste disposal team, right? Now, can you tell me what is the function of lysosome then?
Ma'am, they break down the waste materials which is in the cell.
Yes, Aryan, correct. They carry out waste. Sujal, carry out would not be the right term. They basically what they do is whenever they encounter waste products or harmful products in the cell, they burst open, okay, and they just take over the whole thing and kill it with themselves, right? I'm not sure, you might have heard the term like suicide bombers, right? Similar function, but this does that job inside the cell. So it contains enzymes, right? Yeah. So lysosome contain enzymes which basically help in breaking down the waste products or harmful products, right, in the cell. That's why they are called the suicide bags, because when they encounter something harmful, they'll break open, they'll release all these enzymes outside, and they'll uh, destroy everything around it. Okay. So what happens is, in coming back to plant cells, why plant cells don't need lysosomes is because the cell wall does not let, it offers a very good gatekeeping service where it does not let these foreign particles enter in the cell in the first place. So the need for an organelle which is going to kill them is not there, right? That's why they don't have lysosomes. Okay. Yeah. So for your answer, coming back to our answer, uh, yes. So coming back to our question, you can write. So the name of the structure would be lysosome, and its function, yes, we discussed about, right? So you can write that it is called the suicide bag of the cell, and it basically its function is to capture or release enzymes and destroy the harmful substances or waste products like that. Okay. Then which should we do? Hmm. Okay, this question might be difficult. What are dictyosomes? Can anybody try? Okay, I'll give you the answer for this question since I think we had discussed it when we were doing the chapter. I'm not sure if you would have taken it down. Okay. Right, so this is the question. I think question number 70. Okay. Somebody has tried. Okay, Suhani. It is where the protein is stored in the cell. Okay. Um, okay. So basically, if you remember, there are these this structure inside the cell, right? Can you tell me what is this? What have I drawn? I mean, might be.
Is
Golgi apparatus.
Golgi apparatus.
Yes,
Golgi.
correct. Good, good. So this is our Golgi apparatus, right? So in the Golgi apparatus, you have these. So if you dwell deeper or into the structure, into what exactly the Golgi apparatus is comprised of, you will notice that there are these flat membrane-bound cavities. Okay, would be something like this inside the Golgi apparatus, right? Okay, so these flat membrane bound cavities which make up the Golgi apparatus, right, are what are known as dictyosomes. Okay, so this would be what are they, and okay, what their function is. So function by you can write the function of Golgi apparatus itself, or like Suhani mentioned, yes, they are involved in protein storage, right? Or basically, the main function of Golgi apparatus is packaging, right? So they are involved in packaging also. So, okay, function would be protein storage, packaging, etc. So basically, what they do is they take over the proteins and then they ensure that it is packed and stored and sent off properly. So that kind of uh, function is what they do. Right. So while writing the function, even if you skip storage is fine, but don't skip packaging, since Golgi apparatus is mainly involved in packaging, right? Okay. Okay. Have you taken it down? Should I go back to the question? Okay. I'm assuming you have done. Okay. Okay. Name the energy currency. Okay. This question is another one which you might not be aware of. The 25th one. Plant cells have large vacuoles, each surrounded by a membrane. What is the name of this membrane? So basically the vacuoles have a membrane, right? When we were talking of vacuoles, I had spoken about this membrane. So they are asking you the name of this membrane. Anybody remembers? Okay, I have one answer. Yes, yes, Suhani, that's correct. What about the others? Do you all remember the name? Can take a guess. Okay, so the membrane is called tonoplast. Now, do you remember? Does it ring any bells? Yes. Okay. So when we spoke about vacuoles, I had mentioned, right, the name of the membrane is tonoplast. Okay, so it is called tonoplast. Write it down. I hope you are taking it down, right? Okay. Okay, so that is all for theory based questions. Let's go to give reasons. Okay. Uh, okay, let's do the second one. A cell is a building block of an organism. Explain why. Simple. Can you try? You can type in your answer or you can unmute and answer. Anything is fine. I can give you a hint. The answer would be from the introduction part of the chapter. Okay, I'm getting answers. Good. Yes, Aryan, that's correct. Sujal, yes, now you wrote the point. Yes, Suhani, your answer is also correct, right? So if you remember, cell is basically known as the building block, right? Why is it known as building block? Because everything is made up of cells, right? Yeah, Aryan, not to worry, I understood it was a typo. It's okay. Yeah, so when we discussed tissues chapter also, the first slide which I had shown, right? Yes, Ashka. Yes, so everything is composed of, all living organisms are composed of cells, right? Cells then come together to form tissues, right? Which then come together to form organs, organ systems, and then finally an organism, right? So it all comes down to cells, right? That's why they are known as the building block of an organism, because yeah, they are the fundamental or the basic unit of life, and they, I can combine all of your points and give an answer, which is basically A. It is the, okay, should I write it? I'll write it. That's better. Okay, so first point being it is the basic unit of life, right? Second point being all organisms are composed of cells, right? And what else can I add is because, yeah, cells multiply to form, right? So this is basically the point where I was saying that cells give rise to tissues, and tissues then go on to give organs, organ system, and organism. That tissues flow chart, the first flow chart, right? Yeah, so this is the third point, is basically this flowchart put into words, right? So they, you can write it that way also, that cells come together to give rise to tissues, which then form complex organs and organ systems, and ultimately constitute an organism, right? Okay. Okay, yeah, third give reason is simple. It is a cell theory question. It's a direct question, right? Okay, fourth one is interesting. Why is endocytosis found in animals only? For that, for us to discuss the answer to this question, I want somebody to tell me what is endocytosis.
Ma'am, endocytosis is basically the process through which amoeba acquires its food. Uh, yeah, it's the process through which amoeba and acquires its food.
Okay, yes, correct. Okay, so amoeba, instead of just saying amoeba, I can just, extend it and say organisms, right? Or animals, because in this case they are saying why it is not possible in plants, so let me just say animals, right? So should I write it here? Basically, endo means inside, right? And inside, cell is what is cyto. Cyto means cell. uh, yeah. So inside cell is what it is trying to say. So what inside cell is? Basically the organism taking up food by engulfing it. Yes, yes, Aryan, yes.
Ma'am, may I say it or do I write it in the chat?
uh, the answer for why does it happen in plants?
Yes.
Yeah, you can say, not an issue, you can tell me.
Ma'am, I think it's because plant cells have a cell wall which makes the cell really rigid. And for endocytosis, the cell engulfs the food, so they have to change shapes, right? So it makes it really difficult.
Yes, yes, correct. Awesome. So that's the correct answer. So basically what happens in endocytosis, if I were to show it to you with a diagram, is suppose this is amoeba, right? And this is the nucleus. And suppose this is a food particle. Let me show you with another color. Right? So what amoeba will do is it will eventually, it will form like a, it will extend its body like this, something like this, and it will surround the food particle, right? And in the next step, it will take it up. So now the food particle will be inside the amoeba, right? Okay, so this entire process is what is called endocytosis, right? And now what happens in plant cell is that you have this thick, rigid cell wall, right? As Swani mentioned. And then inside you have this flexible plasma membrane. This thick rigid cell wall outside, oh, let me write it with black. Yeah, yeah. Because it is thick and rigid, it does not really change shape, or it is not flexible. Okay, not flexible, right? So because of this, endocytosis cannot occur in plant cells, right? Because it is not going to be able to bend or move or engulf any food particles or form this arrangement, basically this arrangement right here, right? This is not possible, hence endocytosis occurs only in animals. Okay. Okay. I'm just like explaining the basic idea here. I mean, I hope that you are writing the complete answer in your words, right? If not now, maybe you can write it after class. Yes, yes, Aryan. WBC also eats bacteria using endocytosis. Yes, yes. So that process itself of taking up food or taking up something is called endocytosis. Right? Like I said, my like this is something I follow to make it easier for me to remember later on is by breaking up the words by the meaning, right? So endo is inside and cell is cyto is cell, sorry, right? So something which is going inside the cell, right? Okay. Okay, why are lysosomes known as scavengers? We covered that, right? Okay, this is an interesting one. Again, this would be, I think, a bit difficult to answer at this stage, right? Okay, why do some regions appear darker than other regions on staining the cell with methylene blue? Name the other solutions that can be used for staining. Does anybody would like to try to answer this question? Would anybody like to try? Okay, I have uh, answers. Wow. Okay. Good, good. I'm glad. Okay, so I think everybody has the answer for the second part, is it? Iodine. Okay. Because different parts of the cell take up different colors. Yes. Yes, Suhani. That would be the simplest or the basic way to answer the question. Yes, you're right. Okay. Okay, coming to the technical part, like what is methylene blue and why does it take up only that? I will write it here. Okay, so yes, as most of you are correctly saying, and you might be knowing, methylene blue is basically the name of a dye or what we call as stains, right? So this is what we call as stains in the lab because it stains the cell or it stains whatever you are trying to observe under the microscope. Your first question would be why do you need these, right? Why do you need? It's because when you try to observe anything under the microscope, everything looks the same. You can't really, it's not like a picture which we have shared in the slides or which you see on Google and all, right? So it's not like, it's not going to be that you have, let's say, yeah, let me draw it here. So it's not going to be that this is your cell and then you have like nucleus colored in blue and then the Golgi apparatus like this in different colors, right? Everything looks the same if you try to observe whatever you're trying to observe. Literally everything looks the same. So what we do is we add these stains and particular regions, or we know what, by different trial and error of course, right? After a lot of experimentation, scientists came to the conclusion that, hey, suppose example, this part takes up methylene blue. Or if I want to observe this region or this organelle, it takes up another stain. Okay, let's say it's called safranin, right? So they observed that. So now what do you do is you have a specimen, right? Or you have something, you have a, uh, maybe a drop of blood on your slide, okay, and then you just add the stain or whatever you're trying to observe, you just add the stain, right? So in the cell, in the layer of cells in that specimen, everywhere where there is, let's say suppose a part, you know So uniformly that A part will take up this stain. So when you are observing under the microscope, you can look at it and say, oh, here it is. You know, just for example, if this is the nucleus, suppose the nucleus has taken up the stain. So when you are observing the slide, you can easily pinpoint and say that, oh, look, there is the nucleus. Okay, so that is the whole purpose of stains. Like that, so stains are given names based on how they look like sometimes, or sometimes based on what color they impart to whatever you're trying to stain. Okay, like the name suggests, methylene blue is blue in color, or why, you know, on the sides of a violet blue kind of color, more darker blue is what I'm trying to say. Saffronin is red or reddish orange in color, right? And then you have fuchsin, you have other, iodine is again purplish, violet, some color like that, right? Okay, so coming back to methylene blue stains regions which are rich in, I will write the complete statement. Stains regions which are rich in, yeah, DNA RNA, right? What do I mean by rich in DNA RNA is that you might find DNA RNA scattered everywhere, maybe in your specimen, but wherever there is more concentration, right, that place would appear darker. So more concentration would appear darker, right? So that's what I mean by a rich in DNA RNA, which means if it is there in if it is concentrated in a region, that area would stain darker. That's what you mean by stains darker, right? Okay. So yeah, if you know this much information should be sufficient. Like there is a more detailed approach that which exact part of the DNA or RNA does it bind to, right? That's why we use this, because it binds to that part, right? But for that I need to explain the bases which make up DNA or RNA and all those parts, right? So at this stage, this much information is sufficient. And yes, so other stains would definitely be, as you all have already given me one, I will add the names which are new for you, which would be, yeah, one is saffronin, of course, right? The other is called crystal violet, and there is one more which is called, okay. So depending on how many they ask, I think generally they just ask two, right? One or two if you give is enough. Any names whichever you are comfortable remembering, right? Basic fusion, again, it is a color. I'm not sure if you are into arts or painting, you might have heard this color name fuchsia, right? Yeah, so that same color, that's why basic fusion. Okay. Okay, yes, one. Okay. Paper pattern at the end. Sujal, what I don't understand. I mean, paper pattern as in? Or an example. Uh, how the paper will be set? Okay, that actually depends. So now there is no fixed procedure as such, because last year also it kept changing because everything is online, right? Marks and things. Okay, like that. Okay, usually you have for your first set, first let me discuss it now only. For maybe your FA, you will have simple paper, like few questions or 2 marker, few 3 markers, and few 4 or 5 markers, right? And maybe it depends. So if they have told you there will be a MCQ part, so there might be an MCQ part also, right? Or if not, it might just be a 2, 3, 5 marker question. Based on the type of questions, if you want to know, no, there is no Google form. Then there is no MCQs. Then they want you to write it down. Okay, then no MCQ. Then maybe there could be 1 marker questions also. Again, it depends. So this varies from school to school, and it depends on the the current situation or the current optional. Yeah, right. Okay. But usually in your first paper, it will be on the easier side, and you might just get difficult questions as 5 markers, right? The others will be fairly easy to solve, provided you have read and done practice and then go on to give your exam, right? Yes, then definitely it would be easy. Uh, what else? Okay, so the max marks are 5. Yes, max marks are 5. 4 or 5 again depends. So depends on the paper. You know how much marks the paper is for. However, it adds up. Like suppose if it's a 20 marker, they'll distribute it in such a way that it comes up to 20. So either 4 or 5. Yeah. And like I said yesterday, usually the 5 marker questions are difference between, right? Difference between, in cell you will get diagram, draw a diagram of plant cell, animal cell, right? Okay, you want, I'll write it here. Diagram of plant or animal cell, right? So this is because you are covering 10 marks for biochem and physics each. So yeah, I don't really think they'll ask you a 5 marker question in this exam then, because it's hardly anything, right? Isn't it? So yeah, so they might just stick to 1, 2, and 3 markers. Then for the next exam, it would. Thank you. Somebody has sent the whole thing. Yeah. Yeah, Sujal. Yeah. Okay. So you don't really have the whole chapter also, right? You just have till cell wall, and you have pen and paper. Okay. It would be 30 marks. Yes. Okay. So if it is till cell wall, then there won't be all this also. Then I'll tell you what will be there. Just let me check. Just a second. Yeah. Okay. So I think if it is still cell wall, you might have osmosis included, right? So osmosis is sufficient for them to question you enough, like give you ample questions to answer. Okay, let me just check what is the part till cell wall. Huh. Okay. So yes, so all this is there, right? So you have cell theory, then you have diagram drawing for microscope. No. You might have labeling. Drawing might not really be. Um, I haven't seen a question, at least last year I haven't seen a question where you had to draw a compound microscope. Again, like, because they keep changing the pattern and everything, I cannot really tell you 100% that it will not come. But yeah, labeling could come. So it's better if you have a rough knowledge of the diagram, that where is what is sufficient, right? So these are the 10 things you should know, where is what in the diagram, right? Cell theory is there. So history, yes, they'll ask you one marker question, which is who discovered nucleus, who observed cells, right? First Robert Hooke, then who discovered nucleus, Robert Brown, then who coined the term protoplasm, and cell theory was presented by uh, so these four people, one, two, three, four, right? Leeuwenhoek, I'm not really sure. Yes. There is a part in the, yes, definitely. There is a part in the workbook where you have to draw diagrams and microscope is written. Yes. But uh, at this, I mean, your first exam, I'm not sure if they will ask. Like once you complete the chapter, you might have again an exam, right? They might ask you that time, right? Uh, Labeling could be asked right now. So labeling, do prepare the labeling. Yeah, exactly. So that time they might ask you the diagram. For now, labeling they can ask you, right? 80 marks. Okay. Yeah. So history, you just need to know the basic information. Yeah, this slide is sufficient for history. And cell theory, yes, this question you might get it, which is there here, right? In the theory based question, there is one question, that same kind of question you can be asked, right? This one, see, this GR3 question, something like that could be asked, that all plants and animals are composed of cells. The above discovery was made by whom and what does it refer to? So this is a cell theory question. Okay. Okay. Then coming back to what else you could get. This we discussed, basic labeling, right? Okay. This nothing to ask on this. This also, I'm not sure. They might not really ask you anything. It's a very straightforward question. They have nope, nothing from here. Yeah, this point could come as a fill in the blank, that what is plasma lemma, right? This could come. So just remember what is plasma lemma. And then, no, diffusion basic definition and one example, right? So definition and example of for diffusion. Osmosis is very important. Even if you are skipping some topic, for your exam, please don't skip osmosis. Definitely there will be 3 4 questions on osmosis, right? Considering that they are not, I mean, they are not testing you on the full chapter also. So again, so osmosis will be the highlight on which they ask you questions. So just be thorough with it. So the questions could be anything. It could either be something like, the vegetable wala question we just did, where you're adding salt into something and then what happens. Or it could be just identifying which is a hypotonic solution, or which is a hypertonic solution, right? It could be either of the two. Okay. And yeah, because what is endosmosis, exosmosis also they will ask. Yes, correct. Till cell wall hai na. So this is definite. Diffusion, they might just ask you a what should I say, definition. What is diffusion? See here, there are so many questions on osmosis. I can pinpoint it here itself. This question is on osmosis. Dry apricots are left for some time in pure water and then transferred to sugar solution, right? So what happens when this is done? So for this you need to know what pure water is, what type of solution, sugar water is what type of solution, and then the movement of H2O will be from where to where, right? Okay, this is also an osmosis question, osmosis all. Okay. One odd man out here is this the staining question. So staining actually comes in the introduction part of the chapter. So this question again would be something could be asked on staining, right? In your NCRT, 2-3 lines are given on staining, but what we discussed in the answer actually covers that whole part. So whatever 3-4 lines are given in the NCRT, we covered it in our answer. But still, what you can do is you can read through it once more, and in case you have any questions or anything, you can always ask me or ask on the group, right? Anytime. Okay. And yeah, so in the workbook itself, there are ample of questions on osmosis, right? What I can do is, acha, I'll do that. After class, I'll send you a list of questions from theory based, all this, give reason, what happens when level 1 and level 2. Okay? All questions till cell wall. So, whatever is coming in your exam, those questions I will give you as homework. So you can do that and I mean submit it on the link or form link wherever is mentioned in the group description, right? So that way you will have a proper practice of uh, the questions, right? And it would be like a question bank. So you would know that what type of questions can come, and your practice would also be done. You will be a bit more confident also, right? ठीक है. So after class, I'll give you homework, but in class, let us discuss a few more questions, right? Okay. Okay. Yeah, see all these tiny questions. So they are there as subdivisions also. So if you see, give reason 8 question, the first two might not be relevant right now, but the third one, if you look at it, plant cells shrink when kept in a hypertonic solution, right? Okay, so you might get questions like that. Okay, so what would be the answer for this? Why do plant cells shrink when kept in a hypertonic solution? Okay, what I'll do is I will quickly revise osmosis for like next 10 minutes, because I think again you might have some questions, some doubts which are not getting cleared, right? Because I remember one of you had sent images on the group when we had done the chapter itself, saying that you were confused uh, in the chapter notes, because one place it was written hypo as high and the other place it said dilute solution, some things like that, right? You are getting confused because of those things. So let me. Okay. uh Yes, so Aryan, the phenomenon would be plasmolysis. They are just asking you why does it shrink. So why does it shrink? May you will answer yes. Suhani has answered it correctly. So basically, why would anything shrink is because it loses water, right? Okay, so I'm doing a quick recap on osmosis. Osmosis is basically when there is, uh, you know, okay. Suppose if these are solute particles, I'm labeling it, and I'm going to draw water in blue, right? Okay. And this here, let me change the color. Okay. So osmosis, if you break down the definition, is basically the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane, right? Which basically is this way, if I am saying that this is H2O, right? So if you see there are, okay, let me just change the color completely because, yeah. So this is my solute particle, right? Okay, now it should be better. So now if you see there are like two molecules of water here versus there are four molecules of water on this side, right? Which means the concentration is higher this side. So the water molecules will move from here into this part via the semi-permeable membrane. So only when there is a semi-permeable membrane can you say osmosis has occurred. If it is a completely permeable membrane, or if it is let's say something else, so there is selectively permeable, there is completely permeable, or there is, let's say, impermeable. Impermeable definitely will not allow any exchange, right? No exchange is what you mean by impermeable. Selectively means there is, it has a list, okay, like a list of people or a list of uh, people. I'm just giving a real life example as such. You could consider it like your class list. So when your teacher takes attendance, she only calls out the people who are there in your class, right? So she will not call out students who are there in, let's say, other divisions. So that is what is selectively permeable, that the cell or the membrane has a list of items which can pass through, right? A list of particles which can pass through, the others are not allowed. Semi-permeable is this, that only specific molecules based on the size, right? Based on the size, here the criteria becomes size, that's why it is semi-permeable, right? Only based on the size they can move to and fro, right? Completely permeable means everything can pass through, and impermeable means nothing can pass through. Okay, okay. So that is the basic definitions for membranes, right? So osmosis will only occur when there is a semi-permeable membrane involved, which means when any time you talk about any cell, right? Any cell, there is osmosis involved because we have something called the cell membrane or the plasma membrane, right? Which is definitely semi-permeable, right? Okay, okay. This is done. Then let's come back to the three types of solutions then, which are basically hypertonic, hypotonic, and isotonic, right? Okay. Hyper, like I said, make it, at least for this part, I would prefer if you could break down the words, right? Because it's easy to remember these like this instead of just, rote learning the definition for these three solutions, especially because it gets confusing, right? Hyper means high. Remember it like, suppose somebody who is very excited and is running around excited or screaming, you might have heard people say, usually it's like people who are adults, or they will say this when kids are in a frenzy, or they are running around like uh, enjoying or something, whatever, playing, right? Yes, yes, Suhani, hyperactive. Yes, Aryan. Yes, hyperactive. You might have heard that word, right? People, so you call the kids that, why are you so hyper right now? Correct? Yeah. So if you can connect the two terms, you can connect that hyper basically means, they are like high on energy, right? High on life right now. So connected that way, hyper definitely means high. Okay. So what will hypo be then? Hypo will be low. Okay, if you can remember high, definitely the other one would be low. Acha, okay, Aryan is a real life example of a hyperactive kid. Nice. Okay, so it'll be easy for you to remember, isn't it? Hmm. Okay, yes. right? So if you remember hypertonic as high, the other one definitely would be low. That is my way of helping you. I mean, uh, you know, my way of maybe remembering what is low and high, right? And iso is basically same. Iso means same. Iso you will come across later, like in many other words. You can remember this if you remember this term in maths, right? Isosceles triangle. So isosceles triangle basically has two sides which are same, right? That's why it is known as an isosceles triangle. That's how the name comes into picture, right? Again, if you if you delve more into math related subjects, you might also come across something called as isodiametric. Again, same diameter, you break down the words, right? Okay, so something like that. So isotonic would definitely refer to same would be isotonic. This one is not frequently asked, so you can, it, if you can keep it at the back of your mind. But these two, they keep coming, and all osmosis related questions will have something to do with both these terms, right? So if you could memorize the meaning of this, would be wonderful, right? And again, like I discussed in the first question which we took up today, what is high, right? If I'm saying hypertonic is high, what is the high part I'm referring to? Is always the solute, right? I'm always talking about solute concentration. Okay, solute, you can remember, if it is, again, if it is a term which is difficult for you to remember, think of sugar water. Okay, always sugar water, salt water, anything which you like, right? Remember it like that. Okay, if you have a sugar water, water is our solvent, right? Sugar is your solute, and the whole thing is called a solution, right? Something like that. So remember, sugar water, where sugar will be the solute and water would be the liquid. You can remember just liquid, right? You don't need to remember solvent also at the moment. Okay, so hypertonic basically means high solute or high sugar or high salt, right? Because at this level, mostly questions will come pertaining to sugar or salt only. Like if you are putting a cell in sugar water, what will happen? Or if you are adding salt to vegetables, what will happen, right? So always, so both sugar and salt would be hypertonic, right? Because they are high in solute, correct? If I tell you I am putting it in water, water would be low solute, correct? Water would be an example of low solute or hypotonic. Okay. Once this is clear, we come back to the two types of osmosis, which would be endosmosis and exosmosis. Again, you break down the word here, right? Exo means out and endo means inside, right? So if I'm breaking down the word again, this means the water molecule is moving inside the cell, right? I can draw something like this, right? Versus if I am saying exo out, which means water molecule is going out of the cell. So exosmosis occurs, right? Okay. Again, you can make a rough diagram of this in your notebook if you haven't taken it down before, right? Okay. So once we are clear on this, there is one last part of osmosis which we need to know is, if I am placing something in hypertonic solutions, what happens to the cell? Right, I think when we discussed the chapter, I had given you a small hint about this, right? Hypotonic has an O, correct? So definitely if you're placing a cell in hypotonic solution, it will swell up, right? It will become O. Versus if you are keeping it in hypertonic, look at the same letter after P. It has become O to E, right? So O becoming E means it's become smaller, something like that. So it will shrink. Again, this is like a cheat code, I would say, because I mean this is not how you should remember. But I agree, these terms are interchangeable and they are really confusing after a point in time. That what is hypertonic and why does the cell shrink or swell, or it definitely gets confusing, right? So this is like your cheat code. You should write it in your book somewhere, right? You have this, you are good to go. You not make a mistake again, right? So once you have identified, so now that you know this, your only job is then to identify whether the cell is in hypotonic or hypertonic solution, right? Then you automatically know what will happen to the cell. Okay. Okay, has everybody taken this down? Then I'm going to uh, give you some questions, like play a rapid fire round with you. Should we do that? Okay. Okay, so the concepts are fresh in your mind. I am going to ask you a couple of questions. Just tell me, you have to tell me whether it is okay. You have to tell me two things. One, whether it is hypertonic or hypotonic solution, right? I'll write it here. Tell me whether it is hypertonic or hypotonic. Okay. Second thing you tell me is what happens to the cell. So what happens to the cell is what I want to know, or in short, the answer is, will it swell up or will it shrink, right? So I want all of you to keep, let's play a quick game. You can keep your chat box open, right? Because I want all of you to answer. Just simple questions based on what I just discussed about osmosis, right? So this is just for me to know that you've understood. Okay, keep your chat box on, and I will give you some examples of solutions, right? So this is your cell, and I will give you examples of solutions. So you have to tell me whether the solution is this, and then second is what happens to the cell, right? Both answers together, correct? Okay. Everybody ready? Should we start? Okay, cool. Okay, I've got answer from Sujal. Sujal is ready. Okay, I hope the others are also ready. We'll start. Okay, I'm taking. Yes, cool. Suhani Aska, yes. Let's start then. For starters, we'll take up simple solutions and then I will go on to complex ones. Okay, good, good. Okay, so everybody's chat option is on. Good, good. Okay, so my first question is, I have a cell and I am placing it in salt solution. Okay, I have got an answer from Aryan. Okay, Sujal. Okay, what about the others? Once I get all the answers, I will tell you. I will discuss the question. Very good. Yes. Okay, three people have answered. What about the others? Very good, Samruddhi. Okay. Okay, who's left then? Suhani, Samruddhi. Okay, let me just check who all have answered. Aryan, Sujal, Ashka, Samruddhi, and Suhani have answered. So we are waiting answer from Swayam only, right? Swayam, quickly answer so that we can discuss. Okay, okay. I think Swayam is Okay, Aryan is changing his answer. Okay, let's see. Okay, so I haven't got answer from Swayam. Okay, I'm excusing you for this round. I want the answer next round, okay? Okay, you did. Okay, and what about the second part then, Swayam? The cell will swell or shrink? Tell me. Okay, I have got answers and I'm happy that most of you got it right. Uh, Really, I'm really happy. That means you were not confused with it. Okay, so the correct answer would be that this is a hypertonic solution, right? Like I said, don't get confused with yes, Swayam, yes, correct. Good, you've answered. Do not get confused with yes, yes, Suhani, yes. Yeah, coming back, do not get confused with sugar and salt. I think people get confused because you feel that sugar is the only one which is hypertonic, salt becomes hypotonic. Something like that is registered, I think, in your mind. So, yeah, okay, Swayam. It's okay if it's slow. I just want you to answer. I'll wait, not an issue. Okay. Okay, so this is one thing I want to get it like crystal clear. Sugar and salt both are hypertonic, right? If I tell you anything, okay, anything if I'm mixing dirt and water also, it would be a hypertonic solution. Why? Because the dirt is the solute here, right? The water concentration is less. The minute I'm mixing any solid in water, it becomes a hypertonic solution. You must keep this in your head, right? Okay. Okay, and what happens to the cell? It will definitely shrink. I'm very happy that most of you gave the correct answer. I hope the ones who gave hypotonic are crystal clear about this now, right? So similarly, so I'm not discussing sugar here because sugar and salt both will have the same answer. I want you to like literally carve this in your brain, right? Both are hypertonic solutions, right? Okay, so now we are going to level 2 of our quiz, and now I have a question to you. Okay, again, same, I would like your same format answer, that whether it is hypotonic, hypertonic, and what happens to this cell. Okay, now, but this is a little complicated scenario. Okay, suppose I have a cell, this is my cell, and now I put it in sugar water. Okay, and now I am putting it in plain water. So my question to you is here at this step, what is the solution and what will happen to the cell? Take your time, I will wait, but I want you all to try.
Now my voice is breaking. Could you repeat the question?
Yes, sure. So I said that there is a cell, okay, and I have put it in sugar water, right? And then I take it out of sugar water and I put it in plain water. Okay, so now my question to you is, what is this plain water? What type of a solution is it? And what will happen to the cell in plain water only? Now I don't want to know what happened in sugar water. Okay. Okay, Sujal has answered. I'm waiting for the rest of you to answer. Yes, okay, I've got answer from Swayam and Samriddhi. Okay, Aryan has also answered. Uh, oh, Aryan has given a complete descriptive answer. That's good. Okay. Okay, let's see what the answer is. Suhani has answered. Ashka has answered. Okay, so I think everybody has answered, right? Did I miss anybody? Nope, I did not. Everybody has answered. Okay, very good. I'm glad to receive answers. Let us discuss the question then. Okay, let me clear this. Yes. So my question was, there is a cell. I put it in sugar water, right? And then I'm putting it in plain water or just water, right? And what happens here? Okay, so let's break it down first. So tell me, this solution is hypertonic, hypotonic,
Hypertonic.
hypertonic. Very good. So cell will
Shrink.
shrink. Awesome. Yes, shrink. Correct. Okay. So now basically I have this shriveled something here and I am putting it in water. Okay. So tell me, what do you think at this condition, which one has more water? You know, this shriveled cell or this, which has more water concentration among the two?
From the water.
Water. Correct. Yes. Water. Water. Okay. Very good. Everybody answered the correct one. Yes. So water has a higher solvent concentration, or basically what solution will it be?
Hypotonic.
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 Yes. Yes. Correct. Now everybody got it right. Very good. Okay, so this will be hypotonic. And what will happen to our cell?
Ma'am, it will swell.
Swell up. Awesome. Excellent. Yes. So this is what will happen, right? I gave you this question because mostly you will directly jump from here to here. You might not analyze what happened here, and then you might not compare this state of the cell with the water and then give it the name of the solution, like then call it hypertonic or hypotonic. Okay. So one thing which is very important for you to note at this stage is that these terms, hypotonic and isotonic, are all relative. Okay, they are what are called as relative terms. What do you mean by relative is that you need two things to compare it with. So there is nothing like, there is no solution on the planet, or there is no liquid, or there is no what should I say, component which is hypertonic forever, right? It is a relative term, meaning I will use it to compare two things. So among two things, one of them will be hypertonic, the other will be hypotonic. And if both are the same, it will be isotonic. So these terms are always used when there are two solutions, right? Or one solution and one cell, right? So whenever you are identifying, or whenever you are naming any solution as hypertonic or hypotonic, always check what is your comparing thing. Like here it was this shriveled cell, right? So always look at your comparing factor, that what are you comparing it with. Then you automatically get your answer, that okay, yes, which one has higher water concentration and which one will have lower water concentration, right? Okay. So yeah, again, one more thing, this is very important to note, that these are all relative terms, right? Which means you need at least two things to identify which is a hypertonic, hypotonic or isotonic solution, right? So mostly questions you get will always be like this. Famous one would be the raisin, right? Sometimes they ask you, a raisin is kept in water, right? Okay. So if I'm keeping a raisin in water, let me draw it here. I hope all of you know what raisin is. It is a dried grape, right? You might have it in your dry fruit ka dabba maybe. Okay, yes. Or I think it is called kishmish, right? Yes. So if I am placing raisin in water, can you tell me water is what solution here? Excellent. Yes. So Sujal and Suhani have answered. Oh, Aryan has also answered. Excellent, excellent. Very good. I'm very happy
Ma'am,
with.
can you repeat your question?
Yes, I was asking that if I have a raisin, right, or kishmish, or the dry fruit, and if I place it in water, okay, so then water will be what type of solution? Okay. Okay, so this again comes back to this example itself, right? You can compare it to this raisin is nothing but a shriveled grape, right? A dried grape is a raisin, right? So again, water becomes hypotonic.
Hypotonic.
Huh, yes, correct, hypotonic. So your answer is water would be hypotonic, correct? Because you are comparing these two. But now suppose if I am comparing, let's say, okay, okay, what example should I give? Okay, tricky one then. Suppose now I have placed this resin in water, right? And I take it out and I place it in a new vessel containing water. What will happen? What is that solution then?
Ma'am, can you repeat?
Yes, yes.
Yes, ma'am. Can you?
Huh. I have my resin, right? Then I put it in water, correct? Now I took it out and I placed it in another vessel containing water. What will happen? Okay, think again. Those who have answered, think again. It is a little tricky one. Yes, yes, Aryan, you got it. Awesome. Okay, Swayam, think again. Okay, let me clear this and explain it to you. Okay. So I said I have a raisin, right? Yes, yes, Aryan, correct. Yes, Sujal, I'm explaining it. I'm explaining it here. The sheet was a bit cluttered, so I'm. Yes, Swayam, you got it right now. Very good. Okay. So I said I placed my raisin in water, right? So as you all correctly said, for this, the raisin would swell up, right? So this is what my raisin has become now. Now, my question was, I am again putting it in water. So what would happen was my question, right? Yes. Sujal, yes, you got it correct now. Very good. Everybody is getting it right now. Okay. Okay, in case anybody has not yet understood, I'm explaining it here. Okay, so in this first step, what happens is this is the result of our first step, right? So osmosis usually occurs till a time equilibrium is reached. What does equilibrium mean? Equilibrium means that both sides have equal concentration, right? Yes, yes, Ashka, you got it correct. Very good. So basically, the if this is my resin and this is water, right? Yes, yes, Aryan, yes, equal. Basically, equilibrium would be equal, right? Correct. So if my this is my resin and this is water, water will keep entering the resin till the concentration on both sides is the same, right? Which means when I've taken it out of the water, there is no more chance for water to enter, or like, the resin has swollen up to that extent that it is filled with water up to its complete capacity, right? If any more, if I put, it would burst, something like that, right? Yes, yes, Swam, it is full of water. So it the exchange has taken place till both the concentration in both the resin and the water was almost similar, right? The water concentration is what we are talking of, correct? So now when I am placing this swollen resin back again in water, this makes this an isotonic solution, right? Why does it make it an isotonic solution? Because once this resin got all swelled up in this part, the concentration of water here and here is the same, right? That's why water, in this case, water becomes an isotonic solution, and your answer should be no change, right? Nothing will happen, right? Yes, yes, Swayam, yes. So if you want, you can do this at home also. You can try it for yourself and you can see. Take raisin. I'm sure, I mean, most of you might be having raisin at your house, right? Some dried fruit, right? If not raisin, any other dried fruit, if you have, just try placing it in. Take one and place it in three different bowls, right? Put one in salt water, put one in sugar water, right? And put one in plain water. And then you can observe yourself what happens, or I mean, you can keep it for some time. Definitely it takes some hours, right? 3 to 4 hours, and then notice what happened. And then you can play around. You can take the raisin out of the plain water and place it in sugar water and see what happened. Then this one you can put it here and see. This one here, right? You can keep mixing, matching the combinations, and you can perform an experiment at your house itself, right? Then you can tell me in the next class that, oh, this happened, or something like this happened, etc. Right? This is some activity for you to do. Okay? This will be like a visual demonstration of osmosis also, and it will help you understand the meaning of the terms hypotonic, hypertonic, etc. also, right? So this example, if you could take a note of this, would be great, right? Because this is a classic example where I am telling you water is hypotonic in one case, right? And isotonic in another case in the same example itself, right? So this is exactly what I mean by relative terms, that you are comparing it. So when you are comparing two things, it can be different, right? Okay, good. So what I will do is, yes, so I will not extend the class today. I mean, since we had more of practice, I'm going to give you all osmosis related questions, and questions till cell wall or whatever will come in your exam. I will assign it as homework, right? So what you need to do is you need to write the answers neatly in your book, take a picture, right? Scan it, convert it into PDF, right? You have these, like, I mean, if you know how to convert it to PDF, nothing like it. If you don't, you can just Google it, right? Google image to PDF. You will get options online itself. You don't need to download apps or anything. Just there will be websites where you can convert it to PDF, right? And there is a link in your group description. There is a Google form link already there, saying homework submission, right? So you can submit it there so that we can have a look and rectify your errors also, right? Okay. So once class is done, I will just give me like 15-20 minutes. I will give you your questions, homework questions, and you can do it over the weekend, right? Maximum day should be like till Sunday, finish it. Okay, don't extend it beyond that. Okay. Okay. And yes, when you're solving, as in when you're solving, anytime you have any questions, you can always ask on the group. Or if you feel like you don't want to ask on the group, you can ask me directly also anytime. Okay, we'll be more than happy to help you. Okay. Okay. So I will do that. Yes. Yeah, I think few of you have some meeting, school meeting also, right? So let me not extend the class anymore. Cool. Okay. So do your homework without fail, right? And see you next class. Bye bye.
Bye, ma'am.
Thank you.
Bye.
Thank
Bye,
you,
ma'am.
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 Bye, ma'am. Thank you, ma'am.
Bye
Have a
bye.
great day.
You too.